Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Well, student, in this video, we are going to learn about the rate of the enzyme action, and there are some factors which are actually affecting the rate of the enzyme activity. Let's suppose, students, if there's in a car and if the car have more petrol, then obviously the car is going to move more, like they have the ability to travel more. But let's suppose if there's less petrol, obviously there will be some kind of the, you can say, problem will be carried while traveling. Similarly, if road is smooth, then the car will faster. If let's suppose there is a dig, so like on the road is not well managed, so obviously it will be the problem for the car to drive. So similarly, students, the enzyme, there are some factors which are basically affecting the activity of the enzyme. So let's see these factors. There are some factors which are actually increasing the activity of the enzyme, and there are some factors which are basically decreasing the activity of the enzyme. So let's see these things. So let's start the topic. Basically, student, the temperature. The temperature used to speed up the enzyme's activity. It used to do what? It used to so the temperature used to speed up the enzyme's activity. And how this is happening, basically all the enzymes used to work in some specific temperature, and this specific temperature is known as the optimum temperature. The temperature in which the enzyme is going to work with the maximum rate is known as the optimum temperature. Let's suppose there are some enzymes which used to work in the 37-degree temperature, and they used to work perfectly during this temperature. It means that their rate is mostly the maximum. Their rate is like their working rate is maximum. So this type of the temperature, that specific temperature, is known as the optimum temperature. But sometimes it used to happen. Now listen to me very carefully, students, what I actually want to explain you. Basically, students, the temperature basically increase the activity of the enzyme. The temperature used to increase the activity of the enzyme. Because why? Because with an increase in the temperature, the kinetic energy used to increase. Moreover, when the kinetic energy is going to increase, it's basically help in the acceleration. It used to help in the acceleration of reaction. So what is happening, students? Basically, with an increase in the temperature, the activity of the enzyme used to increase. The reason is that when the temperature is going to increase, the kinetic energy used to increase. And when kinetic energy used to increase, it used to increase the acceleration of the reaction. But when too much temperature is going to increase, like too much, the temperature is going to increase more than the optimum temperature. When the temperature used to increase more than the optimum temperature, then what happens? Then the protein used to lose its structure. Why? Now let me tell you. When we are going to increase too much temperature, when the temperature is too much increased, you can say, when the temperature is increased, now, what is going to happen? When too much temperature is increased, the vibration, the heat energy will also increase. When the temperature is increasing, the heat energy will also. So what is happening, that when we are going to increase the temperature, well, let's suppose the temperature used to increase more than the maximum, like more than the optimum temperature, then what is going to happen? The increase in the heat energy. The heat energy is also going to increase. When the heat energy is going to increase, it will result in the increase in the vibration. Increase in what? Vibration. So increase in the vibration will start the vibration. So when the temperature is too much high, more than the temperature is more than the, you can say, that optimum temperature, then what will happen? When the temperature is going to increase more than the optimum temperature, the heat energy is going to increase. The heat energy is going to increase, and when there is more heat energy, students, then what will happen? Obviously, when there is a more heat energy, the vibration in between the molecules are going to increase. The molecules are going to vibrate with each other, and when they are going to vibrate with each other, the molecules present in the enzyme, they are going to strike with each other. When they are going to strike with each other, they are going to destroy their globular structure, and the protein globular structure is going to destroy. Due to what will happen? The enzyme is unable to work properly, or the enzyme is dead now. Obviously, students, when its structure is going to lose, when the enzyme is going to lose its structure due to the high, you can say that due to the high heat energy, the molecules are going to strike due to the vibration, so that the globular structure of the protein is going to lose, and this is known as the denature. You can say that the molecule of the enzyme actually denature, and the process is known as, like, you can call it denaturation. Denaturation. Basically, student, in the denaturation will make the enzyme totally useless. Might the enzyme is blocked, or it is not able to perform any kind of the chemical reaction. Now listen to me very carefully. Let's suppose this is the enzyme and this is the active side of the enzyme. Due to the high heat, the denaturation occur and the enzyme structure has destroyed. Now what will happen? There is no more active site present in the enzyme which is going to work properly. So now this enzyme basically blocked itself. No, it is no more able to perform the proper function. If you're going to look in this table, you will get that what actually I mean. I'm just summarizing whatever I teach you. Now listen to me. This is basically the y-axis, which is representing the activation of enzyme. Like, you can say activation of reaction, the reaction speed. And this is the increase in the temperature. The horizontal line is representing the increase in temperature. Now you can look at here at 10 degree, the enzyme is basically the rate of reaction is increasing. When it reaches the 20 degree, obviously the rate of reaction increasing, and then 30, and then 37. The 37 is the point where you can say that the reaction is maximum, because the enzyme is working properly. And here is the point that you can say within the increase in the temperature, it is providing the acceleration to the reaction. This is a point. Now enzyme accelerates itself, and enzyme is performing the maximum activity, and it is performing its maximum rate of the work. So this is the optimum temperature. But when you're going to further increase the temperature like 50, 60 and 70, slowly and gradually when you're going to increase the further temperature, the rate of reaction is decreasing, and at 70 degree what will happen? The rate of reaction will become 0. So this is proving that within the increase in the temperature, like you can say that if you're going to increase the temperature more than the optimum temperature, the rate of reaction is going to decrease. In simple, you can say that before optimum temperature, the rate of reaction is directly proportional to the temperature. But after, you can say that after optimum temperature, the rate of reaction is inversely proportional to the temperature. Is that clear to you? Hope so you get that. So let's see from the book what actually they are explaining in the book. You can see that, students, here that increase in the temperature will speed up the rate of the enzyme catalyze reaction, and the maximum, or you can say that the optimum temperature is there in which the enzyme is going to perform the maximum work. When the temperature rises to the central limit, the heat adds to the activation energy, and it will provide the kinetic energy to the reaction, and reaction is going to accelerate itself. But when you are going to increase the temperature more than the optimum temperature, the energy will increase the vibration, and atom of the enzyme, or you can say they are going to strike with each other, so the globular structure of the enzyme is lost, and this is known as the denaturation of enzyme, and it will result in the rapid decrease in the rate of enzyme action because it is going to block the completely. So you can say that the optimum temperature for the maximum enzymes which are present in the human body